So I want you to think about some things. And what I want you to do now is, I'm the type of lecturer that we have to engage with each other. So you have to communicate with me so I know what's going on. And so for a few moments, what I want you to do is allow yourself to be free. Allow yourself to respond in any way as if you were in your favorite room in your house, whether it's the den, your bedroom, the kitchen, or you're hanging out at your favorite restaurant with friends. So I want you to take a moment, listen to some music, and truly respond to it. Don't act like you're in an auditorium, and don't be stiff. Just respond to it. All right? See, the students understand. So hopefully you do feel good and nice, right? Now, maybe the first part, the classical, that might not have been for you, and it might have been for some of you. And maybe the rap, some of you just detest rap like my dad always did, but my generation formed rap. So you can understand that I have an appreciation for rap. But the point is, these songs sent cues to you, and you responded in a certain way. And so just as you respond, cells responds to cues. And they can respond from cues from their neighbors or the outside environment. And cells can jam too. But you have to watch very carefully as these cells jam, because they can move a little bit quicker than you do. So you can see the movement. And as I said, they move much quicker than you would move. And so in society, we have many ways for signaling and communication. And this particular way is what of signaling? How is our signaling regulated by this in society? Remember, I told you I'm a lecturer that we have to be engaged, meaning you have to talk back with me, and I know you know what this is. So how do we, in our society, regulate signaling? This is one of the ways. What is this? I appreciate that. Thank you. You have beautiful voices, better than mine. So the question is, what signal is transmitted? So what signal is transmitted? Go. Well, with this, what action would you take? So it all depends, right? If you are an aggressive driver such as myself, I will look around. I realize I can't beat my chest. All right. If you are an aggressive driver such as myself, I will look to make sure it's safe, but I'm probably going to go. But yelling means that we should yield and stop. But you understand the importance of signaling. And so cells have signaling ways as well. And don't worry, there's nothing wrong with your eyesight. I intentionally blurred this because cellular signaling is very complicated. And how do we begin to understand cellular signaling? Is we just look at one particular area, such as this molecule or these molecules, to really think about cellular signaling. And so with that, when we look at a protein, we look at them because they're cellular workers. And so I want you to take a moment to look at this video and choose, there are two videos, and choose which way that you think proteins work within a cell. You realize this is the last day of classes. So these were two videos of William & Mary students, right? One video was when the William & Mary students and faculty and staff, I should add, broke the World Guinness Book of Records. That was years ago. It's been broken again now. But they were more synchronized, you know, dancing like this and shaking together, all in uniform. And then you have the last day of classes where what's going on there with the students? You don't know, right? They are just, the seniors are having a ball. That this is it. last day of classes, just have 2 weeks of final, and they graduate. So the question to the audience is, which way do proteins function in a cell? Are proteins moving in the same uniform way, or are they moving well? Well, we need to hear you. I can see you, but administration, President Taylor, Dean Conley, they can't see you, so we need to speak up here. So which way do proteins work in a cell? 2? I like how you just like 2. And so if you think about it, you're right, too, that you have proteins hanging out. Look at there, signaling here. So this is the way that proteins actually work in a cell, exactly how students move around during the last day of classes, but you have certain type of communications. So I want to take the time to say, look, hopefully we're going to have a marvelous night. It's definitely a marvelous night to have a brain dance. So please grab and hold on as I take you through my research program about MKSticks. So I want to introduce you first to the protein family that MKSticks comes from, talk about the function of MKSticks, and then summarize its function and significance and tell you about our ongoing research in the lab. And then, of course, make sure I give the proper acknowledgments. So with that, let's get started. What is this? What do you think about it? Yeah, you all is like, no, I really don't want to deal with this tonight. Well, I say to you, you already know a lot that you don't think about, but you already know a lot about this table. So those of you who are English majors, music majors, see some of my business professors and music professors, they're on the front row like, oh no. I say, but you already really know some things. So what is that element that we're pointing to? All right, what's the next one? And if we put 2 hydrogen molecules together in an oxygen, what do we have? See, look at that. Already smart. We went from an element to a compound. Do we like water? Yes. What about this compound? Why no? What is this compound? And why you said no, whether we like it? White, fossil fuels. So you're right, it's carbon dioxide, so we have to think about it. So see, you already knew things about the periodic table. So if I introduce it to you again, we can be happier about it. Now, my lab works with this particular element, phosphorus. And I'm just going to now just say phosphate, and you know a lot about this. Look what all the things it's used for, right? Detergents, fertilizers, pesticides. Now, of course, I'm paying attention to this part. Like, this is why I'm interested in phosphorus or phosphate. And so the reason why is we have these proteins in the cell here. And so this is a protein. And then if we add this phosphate, and we already know it's an element, so if we add it to a protein, this protein is called phosphorylated. What I want you to pay attention to is when you have a protein that has no phosphate group, then it is a non-phosphorylated protein. We're going to add a phosphate group. Then look at what adds it. So kinases, that's a protein. I normally would say an enzyme, but tonight we'll keep it simple. Kinases, this protein kinases, add a phosphate group to a protein and make it phosphorylated. Now, once a protein is phosphorylated, we have another set of enzymes or proteins called phosphatases. The phosphatases does what to this phosphate group? It takes it out, it removes it. So it dephosphorylates the protein. So now we're going to have to learn some things. So what protein adds to phosphate? Kinases. Keep it in mind. What protein removes it? Phosphatases. And if we want to get a little bit more detail, we can actually call these enzymes. And enzymes are mostly known because of their function. So a phosphatase, and it has the ACE, removes that phosphate group. So you got that? You're following me? All right. So why is it important to me? Believe it or not, this is my field. These are the people in my field. And you can see me there looking so scared. But these are the people in my field. And it's important. And notice this. There are more of you in this room than the people who work on phosphatases. So that is very clear. It's just about 180 of us. Well, what's interesting is this guy here, and now I'm flipping too fast. Okay, this guy here, Nicholas Tonks, in 1988, he characterized the first protein tyrosine phosphatase. And this is who I received my training under. So you might say, Shanta, we really don't care about a British guy. Why do we care that he characterized the first protein tyrosine phosphatase? And I would say you should care because the very first protein tyrosine phosphatase that he characterized has been implicated in all these various diseases. And exactly, wow. And then more importantly, maybe you're like me, I hate wordy slides. As soon as I see a lot of words, I'm going to turn my head away from the slide. That this might bring it to home for you. That, okay, as scientists, what we have is we work with mice in the lab. Well, I don't have any in the lab right now, but a lot of us work with mice in the lab. And I would say to you, imagine if someone fed you a high-fat diet. So for the students at Women Married, that would probably be Chick-fil-A and Qdoba's on campus, right? Your high-fat diet. For faculty, it'll be Blue Talon and what's the other restaurant? But you understand where I'm going with this. Now, which one do you think? So these mice were fed a high-fat diet. And then, do you see a difference in these mice? It was like, are you kidding me? What's the difference? One is, this baby is huge. So what do you think is wrong with this mice? It's fat. You want to be politically correct. It's fat. So I'm going to say to you that this mice is the normal mice. Now, this is the lab mice where the first gene that Nick identified, PTP1B, that gene was removed from this mice or knocked out. So it was knocked out. It wasn't expressed in this mice. So without PTP1B, the mice can be nice, slim, right? But if PTP1B is in this mice, it's fat. And so what are the implications? What do you think about this mice? It's fat and what else? When we think about diseases, you see it all the time in commercials and certain news programs. That what do you think we need to be concerned about this mice? Heart disease? Diabetes, right. So we have to think about that, obesity, heart disease, and diabetes. So that's why we really should be paying attention to phosphatases, that it's going to have a huge role in our society. Diabetes, when you think about type 1 and type 2, are an epidemic that we have to think about. So with that, I want to point out that just next year, our field will only be 30 years. It will only be 30 years ago that the first PTP1B was characterized by Nick. So keep that in mind as we go through this talk. And for here, these are many other protein tyrosine phosphatases. So it's more than just the one he characterized, but there are many. And what I want you to pay attention to is, you don't have to know all these phosphatases, but I do want you to know that if you were to see some alphabets, which a molecular and cellular biologist and a biochemist, such as myself, call amino acids, if you were to see some amino acids, or keep it simple, some alphabets in this order where you have H for histidine, C for cysteine, 5 amino acids in an arginine, or 5 letters in an arginine, when you see this sequence, that is a phosphatase. It is a protein tyrosine phosphatase. It is no denying it, that it is this. So are you following me thus far? All right, you're with me. So with that, it's time to take a test, a short quiz. So you have an insert, and what I want you to do is see, do you have a phosphatase? And if you have a phosphatase, put a rectangle around it, and the first 4 people who thinks they have it, or whoever is the first 4 who has it right, we have a gift for you. So raise your hand if you have it. I kind of want to say students who are in my class, you're not allowed, but I can't do that. Do we have 4 yet? You have 1, so yep. All right, so it's up to the mic carriers to check. Are we ready to check? Do we have 4 people? Okay, 1, 2, 3. All right, so we have 4. And so there's the answer. If you circle that, or I'm sorry, you put a rectangle around it, then it's correct. So now you know how to look up whether a protein is a protein tyrosine phosphatase. So I'm going to move on unless you're enjoying yourself for time, but I really prefer to move on. Okay, so thank you for your participation. So with that, you now understand what is a protein tyrosine phosphatase. You know how to look at a sequence and pick out if it is a phosphatase. So now I'm about to ask you, what about if it's a pseudophosphatase? And I know some people's like, I cannot pronounce that word. The word is too big for me. I'm done. And I say, no, let's back it up. What if it's pseudo? What does pseudo mean? False. So you already know what a phosphatase does. What does a phosphatase do? It removes the phosphate. So if it's a pseudophosphatase, what does it do? It cannot remove the phosphate. See, you're so smart. So it cannot remove the phosphate, as this is showing. But the question is, what does it do? What is its job? And that's what my lab is, has to think about, that what is the action of pseudophosphatases? And the reason it's important is because our human genome, there are 10% of pseudophosphatases within us. And that was kind of subtle, right? You didn't see where that 10% came up. Did you see the yellow to the blue? Oh, you did? Because I was going to ask about 10% of you to stand up in the room so you can really understand what 10% means. That 10% is a large amount. That was if I were to ask this entire front row to stand up. And you know, let's do that. Exercise your legs. Some of you are tall. You're not short as me. So all of you, meaning all of you on the floor, let's stand up, not just the front row. So that's like saying, and I'm sorry to the admin, but that's like saying that they are insignificant. They are phony, fake, they just don't exist. We should not pay attention to them just because they have pseudo in front of their names. And President Reebler is like, it's okay. But okay, thank you for participating. Please have a seat. But the point is, of course, they're not significant, right? You're an individual just like the people who are behind you. And so, as a scientist, when we look at, especially those who look at pseudo enzymes, we understand that we can't avoid pseudo enzymes, that we need to understand them more. And that's the job of my particular lab. And so the question is, what do pseudophosphatases do? And right now, we know that a phosphatase is going to do what? Right? And so pseudophosphatase, yes, it can grab that phosphate, but you can't take it off. So maybe it serves as a dominant negative. And you're probably like, what? In essence, maybe it just serves as a what? A blocker. You know, that maybe it blocks the protein tyrosine phosphatase to prevent it from removing the phosphate group. And that's maybe. I'm here to tell you that it does so much more than that. That it's not that boring old just blocker. It can be, but it does so much more than that. And for me, what's been interesting of working with a pseudophosphatase is the first pseudophosphatase identified actually have a relevance to Styx. And it was intentionally named after the Styx River. And so what do we know about the Styx River? Provost Halloran, you are not allowed to answer this question because that is his expertise as a Greek mythologist. So what do we know about the Greek river or the Styx River? It's in Greece? See, this is a historian. He's going to like, look, it's in Greece, it's real, it's in Greece. And I want the ones in the Greek mythology, you know, where our imagination and creativity, and I know you have that as well. So it is in Greece. What else? Hades, underworld. So what is in the underworld in Hades? But what else? Dead people. What about dead people? And see, now she's going back to the historian way, and she's right. You have the Styx, Euphrates. Help us out, historian. But also, isn't it in Greek mythology that it allows for immortality? Maybe I should have had you help me, Michael. Yeah, we have a beautiful story, right? What do you know about Achilles' heel? So Achilles heals, right? So Achilles was born just like us, but his mother wanted him to have, what, to be immortal, live forever. So she dipped him in, she held him up and dipped him in the Styx River. But because she was holding him, what didn't the river touch? His heel. So every part of Achilles was immortal. And how did Achilles die? An arrow shot him in the heel. And so now when we have pain, or we'll go what? A weakness, I would say not pain, but weakness, we go Achilles heel. So that's the relevance of pseudophosphatases having relevance to Greek mythology. That was great for me because I loved reading Greek mythology as a child. And so the second phosphatase that was identified was MKSticks. And so Styx was the first, and my lab focused on MKSticks. And so this protein has two domains, your dual specificity domain, where I'm just going to say the phosphatase domain. And you already know the sequence for the phosphatase domain. But notice, what's different for MKSticks? Do we see this histidine? Cysteine? Yay, no? Thank you, no. So that lets us know that this is a pseudophosphatase. It doesn't have that sequence that allows it to be protein tyrosine phosphatase. And this particular domain here, this is as a docking domain. And now we live where some of us actually live on the peninsula, or West Williamsburg is near the peninsula, so we understand what's docking. What's the role of docking? Right, coming into port. So basically a protein able to come and dock here. You know that this is the docking domain. So certain proteins are able to bind this particular part of MKSticks by docking, and as Hermine said, coming into port. So and it's my lab's job to figure out what's the role. So just to slow it down, to recap, this is a phosphatase, so it's going to do what? I know you're tired of it now. The other sequence prevents it. And then it has relevance to the River of the Dead. And so my overarching question is, what is the function of MKSticks? And believe it or not, what gave us the function of MKSticks is stress. Now, my mother is probably stressed, figuring out, what is my child going to say? I'll behave tonight. But I know Steve was stressed yesterday when he didn't see me until 30 minutes later. But what happens if you're stressed? What happens to you if you're stressed? Blood pressure rises. What else? Heart rate. Can you function properly? No. And basically, cells do the same thing. When a cell is stressed, so let's start with a normal cell. Normally, a cell has this system. We're familiar with DNA. So it has this system where it can replicate the DNA. The DNA is transcribed to RNA. The messenger RNA is translated to protein. So when the cell is on a normal day, when everything is fine with the cell, you have this messenger RNA translated to protein. But when it's a stressful day for that cell, what happens? This messenger RNA is stalled, and they aggregate together, and it forms what we call these stress granules. So when the cell is stressed, it has a protection mechanism, and it forms these stress granules. And so that is what I'm about to show you, that if you look at this picture, and we're looking at the top, because the bottom are all nice controlled cells. This is just cells, the nucleus and the cytoplasm. But if we incorporate a protein called G3BP that it can stress the cell, if we put this cell, G3BP, this protein in this cell, G3BP, it stresses the cells and causes those stress granules, that aggregation of messenger RNA. So do you see that this is a small green dot here? And then this large globulars, because that's where a lot of them accumulated. Can you tell that? Right. And I should say that this is the green fluorescent protein. We just tag proteins with the green fluorescent protein that we take from jellyfish. So we just tag them so we can visualize them in cell. Now, you can see that this is a globular. In the presence of MKSticks, do they look the same? What does it look like? It's broken, it's smaller. Man, you all are already scientists. You don't need me to stand in front of you, right? So what's interesting is, what's beautiful is that even if we put MKSticks in a different set of cells, and now I'm showing you a different set of cells, you still see these granules, right? Here, here. Now, of course, this panel is MKSticks. What do you expect to see in this panel? Granules or no granules? Exactly, no granules. And so you don't see any granules. MKSticks decreases stress granules in HeLa cells. Now, I need to take a moment to talk to you about HeLa cells because 66 years ago, Henrietta Lacks died exactly this day, 66 years ago, with cervical cancer. And the reason we're able to even do research as science and produce drugs for medicine is because she had an immortal cell line. That the cells that the doctors at Johns Hopkins obtained from her cervix were actually able to grow. They're still growing in some professors' labs, such as mine and Dr. Allison's lab to this day. We still are utilizing her cell to push research in medicine forward. So I wanted to make sure that we understood where these cells actually came from that we're still using today. And it was amazing to realize that it's actually exactly 66 years ago that she passed away, but her cells from her cervix are still growing, and we're utilizing them in science today. And Oprah did make a movie about this earlier this year. And so for my lab, it's been very helpful because it allowed me to create a model for stress granules. And basically, this is showing that MKSticks decreases stress granules no matter what situation that we cause the stress granules. The MKSticks actually inhibits it and decreases it. Therefore, we call MKSticks its toothless regulator. So this pseudophosphatase is able to be a regulator. Why is it significant is this was the first report on MKSticks as the functionality of MKSticks. So we were the first to report this, that this pseudophosphatase actually has a function, and it's that it decreases stress granules. And then the second report, as you can see, are with undergraduates. Elizabeth's from Hinton, and then Michelle and Jamie are William & Mary undergraduates. And so they are able, and notice Jamie is the first author, not me at all, that we're producing this data is with undergraduates who are the leading authors in their own field, because we're leading the way. So at the time that we published this paper, it was also stated that G3BP, that protein that causes stress granules, also has a role in neuronal development. So what do we mean by neuronal development? So think about a neuron. This is a neuron. And we have the cell body here. We have the axon there. And then we have the dendrites. Now, what do you notice the difference? This is a cell body. This is an axon. What are the differences between the axon and the dendrites? Branching, right? So the axon is just this long extension of the cell. And then you have these dendrites where you can see branching. So that's your distinct difference. You know that now. So one of the questions was, does MKSticks has a role in neuronal development? But we didn't use neurons. What we used were adrenomedullary cells. And basically what we're saying is we use cells from a rat that came slightly above from the kidney, and that's what we use as a model. The reason why, as you see here, that PC12 cells, actually they're in the lab, they are these boring cells. They're just round, boring cells. But if you add a stimulus such as nerve growth factor, they become these cells with extensions. And let me go back, with extensions here. So they actually have these extensions. But if we add a different stimuli, you don't see the extensions. So with the nerve growth factor, you actually see these particular extensions. And so that's the reason my lab used these cells, because you can tell the difference between a stimuli that will form these extensions that we call neurites. So with that, the question was, does MKSticks induces neurites or differentiation? So I'm going to show you. These are the normal cells here, nice and round, so I might have that. If we add MKSticks, what happened? So then what's the answer to the question? Exactly, MKSticks does induce these outgrowths. And this is just showing that it is MKSticks alone that induces outgrowths. So with that, I want us to pause a minute. And it is Halloween. So, well, near Halloween. So you think about everyone want to make costumes, skeleton, different things. But most importantly, you understand how our skeleton system serves as the framework for who we are and support, right? Where cells also have a system that's the cytoskeleton system that is required for cellular support. And so let's actually take a closer look at that. We have here the nucleus. We have tubulin in green and actin in red. So you have all these different molecules. We understand nucleus, the round. That's where your DNA is. You have your microtubule, which is a cytoskeleton protein in green, and you have the actin. So you get that, right? And this is important. These are important in neuronal development. So with this, we now have what we call our growth cones. So the green is what? And the red is? Right. So you have the green is the tubulin, the red is the actin. Then look at how the tubulin is long, and then you have the red actin filaments here. And this is called a growth cone. And so this is where you have the start of those extensions growing. This is required for those extensions. So with me saying that, look at MKSticks. You can actually see we have the microtubulars and then the actin. And this is basically, and do you see that? That we have this green and then the red. It looks more distinct than in the normal cells, but in the present MKSticks. And so this is to show quantitatively that MKSticks increases the growth cones. So we know that MKSticks is increasing growth cones in PC12 cells. The question is, we want to know how. So we discussed that PC12 cells differentiate or have these extensions. And if I give you an external stimuli, what external stimuli, whereas two now, that actually causes the PC12 cells from to be round to go to the extensions? What external stimuli? Right? Nerve growth factor, MKSticks, and I'm about to put another molecule in the equation. Also, Rho A is a molecule in the inactivation of that. So I want you to pay attention, because you knew NGF, MKSticks, but let's pay attention to this Rho inactivation. Now we understand, you have it in your mind, Rho A inactivation causes extensions. Are we all on the same page with this? All right. No? We'll talk to me later and we'll catch up. All right. So with that, which one of these represents Rho A inactivation? The top. And I made it easy to you. Red means stop, right? And you would be correct. So when Rho A is inactivated, we have extensions. When it's active, it stays round. And so Rho A is important because this is a molecule that actually helps with reorganization of the cytoskeleton of those tubulin, well, the actin, really, the actin molecules. So with that, when we put MKSticks in, and people say, if you see space between the two, we see space, so we know it's great. But I'll take it a little further. This is the normal control where we see Rho A is higher, but when we put MKSticks in, Rho A is decreased, right? So MKSticks is actually inactivating Rho A, which says it have a role in Rho A signaling. So you might think, what is Rho A? Well, how did you get here tonight? Some of you took 60. Some of you took Route 199, Interstate 64. I don't know if my mom them came, Interstate 95. But you took a certain route to get here. And most of you took a route that you were comfortable with, that you knew. For me, it depends on what the traffic is like or what I'm going to take to go in and out of Hampton to Williamsburg. So, because it doesn't matter to me. So I'm like not specific at all. But I want to bring this up is, let's imagine that, well, not imagine, cells have certain pathways that they have to follow too. And a particular Rho A pathway will determine these particular outgrowths. So let's make it more back to the cellular point, that yes, this is a route, but from the cellular perspective, what has to happen? Rho has to activate ROCK, a molecule called ROCK, which has to activate limb kinase, and this is a kinase, so I can ask you another question. What does limb do? Because it's a kinase. Add a phosphate, right. And then this limb kinase then activates cofulin, and the question is what happens? So as you see here, what's this? This is our nice phosphate again. And you told me to add some phosphate. Now I want you to pay attention to this, and this is important, because I am a biochemist, so I can't just leave here and only have you see beautiful pictures and not see bands that we become excited about. Sorry, Jeff. That we become excited about. But notice, we have coflin. There's no phosphate, so it's unphosphorylated. We have coflin with a phosphate. So coflin actually regulates actin. Remember that? This is actin again. So when coflin doesn't have that phosphate group, what's happened to actin? It chops it up, right? And so I want you to imagine, as we think about Halloween, and you think about whether you're going to purchase a costume or make a costume. I say make your child a costume. They might win the award. And I know my mom remember when I won, when we finally made the costume and not buy it. But when you go and purchase a cloth, you cut it up, right? And then you put it back together for the costume. So imagine that. Well, for actin, at the beginning, actin will be chopped up when coflin is not phosphorylated so that it can be reorganized the way it needs to be, so that then when coflin is phosphorylated, this actin then now can be stacked and stable. And why would it need to be long like this? Because of the extensions, right? You're thinking about the extensions popping out. So keep this in mind now. When you see no band, then this means actin is being severed, so it can be arranged any way it needs to be. But when you see a band here, that actin is stable, and we think about the extensions. So just to focus on this, MKSticks at 0 minutes, whereas the cells aren't told to become neurites or to form extensions, you don't see any band. And believe me, as biochemists, we get excited whether we see band and then no band and band, or band and it's decreasing. We're very excited. We have bands. And you can talk to some graduate students and undergraduates. They'll tell you this is exciting to us. But at 0 minutes, you see no bands, so no phosphorylation. So what does that mean about coflin? Or actin, really? It means that actin is being rearranged, that it can be rearranged. And then here, that now it can be stacked. And so then, what is it saying? That MKSticks, when there's decreases coflin phosphorylation at 0 minutes, so actin can be rearranged. And then at 24 hours, it is increasing it. So now we have this stability where you see these neurites. And so that's the basis of that. What's interesting is I'm not a neuroscientist. I am a cellular, molecular, and development, well, really cellular, molecular, and my last training in biochemistry. So if there is someone who's a neuroscientist in the room, they will tell you, but what does that matter? Those are just cells from above the kidney of a rat. They're not neurons. They're not primary neurons. So what do they mean? That these cells didn't come, from the brain. The question is, what happens if we took the brain of a rat or a mice and looked at the neurons? What does MKSticks do? So does MKSticks have an effect? And this is what you can see. So let's go back now. Here, remember, this is a neuron. This is a neuron taken from a rat's brain. What is this? Yeah, but you know the specific name of the extension. Thank you, axon. And what are these? Dendrites. And this is normal. What happened in the presence of MKSticks? It shrink. Can you tell what's an axon or a dendrite? No. To me, this is exciting that this made a difference. Like, this is really exciting that we had this normal axon. You put MKSticks in the present. Some of you said shrunk. What else do you see differently? We can't distinguish the axon and dendrite. What else do you see differently? Right, these little pits, which are dead cells. So MKSticks might be increasing death, which we call I can't pronounce that, apoptosis. That's funny, apoptosis. But that is what, things that we see. But you can see that definitely it's having an effect. And so if we look a little further, if we start and see, this is the neuron again, axon dendrites, and you have other axons bonding, if we actually look here, we have dendrite there in the axons that it forms what we call a synapses. And the synapses is important for carrying information from one neuron to another neuron or another cell. So let's take a closer look that with this dendrite here, that these dendrite has these other molecules here, these circular molecules or parts of it that are important for carrying this information. So I want you to look at that you have this dendrite, or here, and then you have what we call the spine. What does it look like is happening in the presence of MKSticks? Right, it's longer. And so this work has been so important that it has led to this publication with, your green button is really, all right, killing me right now. That it led to this work, sorry, bear with me here. It led to this work, I'll get it. It led to this work with 5 undergraduates, and my master student would tell you she's my favorite master student because she's my only master student. For now. But it led to work of all of these are, and I pasted them in the order of their status of the publication. So it led to this publication. We were the first to publish that MKSticks, a pseudophosphatase, have a role in neuronal development. And then it led to this paper, and you don't see the student, because if Arya, you can step up here with me, because she's here tonight. This is who I call my mini-me, and that's because Arya, well, I can at least give you a hug. So I'll say this is because Arya reminds me so much of myself when I was her age, her personality, her zest, everything about Arya, except she's so much brighter, so much better looking. Reminds me of Arya when I was her age. And I miss you dearly, actually, in the lab. But this is Arya's, thank you, Arya. So this is her publication, this first, this is her publication as first co-author. And notice she's the only, it's just me and her. So Arya actually did this work, for this figure for this paper while I was in Toronto, and we would talk on the phone, you know, at least 3 times a week. But she got the work done, and so this paper was published. So thank you so much, Arya. And also, the next publication, Arya is also on, but we now have another new student from chemistry, Christina, and you can see some of the same players. Christina, who's also on this work, that's due, you can see this month. Hopefully, I'm going to have it in by next week. But that's what I showed you have led to all of this work. And so the question is, why should society care? Or really, why should you care? And the truth is, you should care because from the very beginning, we already have our genes and certain proteins that may be very relevant and as we age, and it's still a puzzle to us. So we have to go back and really figure this out, because what happens in early development is a puzzle. How do we get here? So we definitely should care. And for me, it became very personal, right? Because these are my maternal grandparents, and they helped my parents raise me, and each one of them passed away from one of those diseases on the list that you saw in the beginning. But what was really quite interesting is this is the day I received my PhD, and this is Granddad. And in order to stimulate him that day, because he was just sitting down, I decided to jump in his lap like I used to do as a child just to get a response from him. Because he, at the time, he had Alzheimer's, and he actually lived 8 years from this picture. Let me see, what year was that? Yeah, about 8 years later after this picture is when he passed. And right before I became faculty, I went home with my mom to help with the care of him. And what we used to do sometimes, because if you've ever been around someone who has Alzheimer's, sometimes it's like they're not there. And it was like, I want him to remember every part of his life. So what we used to do is play his favorite song. and it will always get a response, and he will always stop and look up and pay attention and smile. And so, of course, you know music is used as therapy for many things. And music is used for therapy for Alzheimer's. Scientifically, I think we're still trying to figure out if and what other molecular mechanism, how these certain type of neurons might be stimulated. But it's something for us to really think about. I don't do that research, but it's something to think about. And, you know, even if it's not Alzheimer's, you have to realize we all have a brain. And we all might be confused some days. And we all might, you know, be like, look, I'm delusional right now. If I don't eat, I'm definitely delusional. But you get the point that this is actually important because all of us may be impacted. And when we think about these things, you have to think about there could be a mutation in cellular signaling that might, doesn't mean it will lead to these diseases, that might lead to these particular diseases. And then you think about the potential for therapeutics, and that's important because millions of lives are impacted, you know, by certain neurological disorders. So just to take a moment, we're almost ending about our ongoing studies. If you would take your pipe cleaners and shape it in the shape of a music note for me, please. And you can do it however way you want. You can take them loose, or you can leave them wind together as a triple. It looks like, I guess, a triple helix like a collagen. Dr. McLendon's looks good. So we have this music note. Actually, you did a very impressive job. Now, making an axon. It's funny, some of you are like, my music note is too gorgeous to change into an axon. And you're like, no. So what has to happen to making an axon? You have to straighten it out. What else? So you have to straighten it out, split it, but you have to reorganize it, right? So you have to reorganize it. And the reason I wanted you to do that is because you have to think about what a cell has to do with certain proteins, cytoskeleton proteins. They have to reorganize and shape to form these neurites and extensions and various other parts of it in a cell. And right now what my lab is looking at, if you look at the first Styx pseudophosphatases that were identified, these are proteins on the side, and these circles represent interaction. There are certain proteins that only interact with Styx. And notice there are certain proteins that only interact with MKSticks, right? Well, guess what these proteins are? These proteins are actually cytoskeleton proteins. So my lab now is trying to identify the interaction of MKSticks with these particular proteins that only MKSticks interact with. And then the other ongoing project in the lab actually has to do with CRISPR. And if you're paying attention to Jennifer Lopez, she should have had a pilot NBC movie named CRISPR of gene editing. That was the newest and hottest thing. In the scientific field, we actually thought that some scientists would probably win the Nobel Prize for it sometime this week. But basically, it said, what do we do if we delete MKSticks? We've been putting MKSticks in the system, but what if we delete it? So with that, these, remember, stress granules, MKSticks does what to stress granules? Right, removes them. So if we take MKSticks out of the equation, in the presence of G3BP, should we see stress granules? There you go. Isn't it beautiful? There they are, the granules. So now we're working with CRISPR tools in the lab to actually figure out what's going on with MKSticks. So with that, I want you to just take a moment to see that this work on MKSticks really just started in 1998. And look how far we have come. But most importantly, from my perspective, look who's leading the field. It's William and Mary. Therefore, it's your community. And what's interesting, understand that this is William and Mary. These are undergraduates against places like Cold Spring Harbor, Van Elden, that's in Michigan. These are major research institutions, and even in Belgium and all the different places. So I would say William and Mary definitely should give itself an applause because we are actually leading the field in this particular thing. And so, and with that, I want you to think about this. This is research, right? I showed it to you, well, I tried to show it to you without this clicker going back and forth on me flawlessly, but it appears that everything works so perfectly. No, it's a winding road. And then you think you're there, and then you realize you're not there. And then finally you're there enough to publish, and everybody celebrates science. It's great, right? And hopefully you have had fun with me tonight, and you've been patient, but it is fun, and we should be more engaged. A scientist isn't the only person that has to appreciate science. You can appreciate science, and hopefully I have done that for you tonight. So also I want you to think about change. So change also is this winding road, and we get there. And then we go back and we get there. And just because we have this star, we're not there. We're still going. And the reason I want us to think about change is because we are at a moment right now at the College of William and Mary, or William and Mary as we want to say. We're at a moment at William and Mary where we can celebrate some things. And so one of the things we need to think about is we have two historical moments at this institution that we really need to start paying attention to. Next year will be the 100-year celebration where 24 women walked on this campus and made it a co-educational campus. So that's... And so that's 2018 and 2019. But currently, we need to celebrate right now the 50th anniversary . And actually, the ladies are here where we have desegregation of the campus. And so we have Lynn Brawley. You can just stand up. We have Janet Brown Striford. And then we have Karen Eli, who graduated from biology, the department that I'm in. And so I say this because these ladies are very humble. You know, they are pioneers, and I understand them, you know, being humble, the humility that pioneers have. But I do appreciate William and Mary for standing up and celebrating your pioneers. I think is very important. And I know that you say that you were just doing the right thing and coming to school, you were going to school. I say to you that it's more than that. It is a 50-year legacy that because you were bold enough to go to school and be a pioneer and be the only person that I'm able to stand here today and show you your legacy, that if you weren't here 50 years ago, I'm not here. So I hope you truly understand that and why we want to celebrate you. And with that, as I begin to end this talk and show you all my students that understand they are my legacy, therefore they are your legacy. And I say that to you because people think I'm young. And as you see from this picture behind me, I'm not that young. I already have students who are hosting each other, let me stand out of your way, who are hosting each other at PhD programs. And then if you look at these students here, look at the diversity of these students who were in my lab this last academic year. And I can tell you, it is a challenge, but the most amazing award to work with a diverse of students, because you have to learn to respect each other's cultures. So it's been, you know, and I'm sure they still keep in contact with each other to this day. And then this doesn't even represent the number of students who I have trained at the College of William and Mary, which is still your legacy. And this is just 6 years we're talking about, because I've been holding off lately. But this is just 6 years, right? And then they graduate, as you can see, and the process begins again. So now I'm going to introduce my new bunch here. Andy Matai, if you could at least stand up, come to the front. Ine, Ben, Kirsten, Ashley, Greta. So as you can, and so these, and they have been a lovely, and they are a lovely crew. So as you can see, your legacy continues. So 50 years ago, that was very important. You know, just like, as I said, that when Henrietta Lacks died 66 years ago, of course her family didn't want her to pass, but it's very important. So I say to you ladies, I appreciate you, and thank you so much. You can sit back down, I appreciate it. I appreciate you, and thank you. And so with that, I need to acknowledge, there are many people to acknowledge, but I definitely want to acknowledge Liz Allison because she was the one who recruited me here in the biology department because they were the ones who were willing to make that change. And so here we are now. So I do thank you and I appreciate you. And I know that we're going to always have many struggles and changes because that's how we grow, but we'll do it together. And then I want you to pay attention to this list here. Notice my major funding ended in 14. And so and my lab has not slowed down. And the reason why is because of private donations or private funding and these donors. These are very important to us as researchers. And the reason why is because you have to understand, 20 years ago, or really just 15 years ago, so the young one of us, we waited too late to become scientists, funding was at 33%, and now it's at 12%. So you understand the challenges that we have, but we keep doing this because we love this. And I mean, to introduce students to science is one of the most amazing experiences, and that's why I can stand up here and smile and be happy. And with that, I would say I open the floor for questions. Thank you.